Welcome to The Explainer. Today, we're diving into something pretty wild. We are actually uncovering the hidden molecular secrets of the food sitting right inside your pantry. We're going to explore the famous Dr. Chem experiment on the detection of starch in food samples. But hey, we aren't just doing a standard dry chemistry lesson today. Oh no, we're treating this like a true crime investigation. We're going to profile a chemical suspect, run a full lab interrogation, and figure out exactly how your food is behaving on a microscopic level. So, let's jump right in.
So ask yourself this, is your honey 50% fake? I know, it sounds like total clickbait, right? But food fraud is a massive, very real-world problem. Unscrupulous producers will constantly take an expensive product, think pure honey or dairy like milk and cottage cheese, and just bulk it up using an unmarked, basically tasteless filler. And the most common culprit? Starch. Why? Because starch is incredibly cheap, it looks like a harmless white powder, and it blends right in. But as food detectives, we actually have a foolproof way to catch them in the act.
And we don't just have a way to catch them. We have a method that is insanely precise. The chemical test we are exploring today can detect starch adulteration in a sample of milk at limits as low as 0.02%. That is practically nothing. It means even a microscopic trace of cheap filler literally cannot hide from our chemical interrogation. It's a phenomenally sensitive tool, and honestly, the best part is that the underlying science is super accessible.
All right, here is our investigation dossier for today. 1. What exactly is starch? 2. The detective's toolkit. 3. The experiment procedure. 4. Observations and results. And 5. Real-world starch applications.
Section 1. What exactly is starch? Profiling the suspect.
At its absolute core, starch is a polysaccharide. Remember, poly means many and saccharide means sugar. It is a complex carbohydrate, a natural polymer made up of a long chain of glucose molecules all linked together. In nature, plants use photosynthesis to create glucose for energy, and then they package that glucose into starch to store it for later. Whether you're looking at a potato, a kernel of corn, or a grain of wheat, that starch is essentially a biological battery. And, it serves as the absolute primary source of carbohydrates for us humans.
Now, starch isn't just one monolithic thing. It's actually a mixture of two very distinct molecular structures. Amylose makes up about 20 to 30% of starch. It's a linear chain, sure, but it doesn't lay perfectly flat. It coils up into a three-dimensional helix, exactly like a spiral spring. Then, the remaining 70 to 80% is amylopectin, which is highly branched, almost like a complex tree forming crystalline shells. But I want you to focus entirely on amylose for a second. That spring-like structure with its hollow core is going to be the absolute key to cracking our case. Keep that spiral shape locked in your mind.
Section 2. The detective's toolkit. Time to set up the lab.
Every good investigation needs the right gear, right? We're going to need our suspect food sample, maybe that's milk, a questionable jar of honey, or honestly, just a piece of potato if you want a guaranteed positive control. We also need test tubes, a stand, some pipettes, and for the advanced stage of this experiment, a water bath and a Bunsen burner. But the true star of the show here is Lugol's iodine. This is a specific solution made of 5% elemental iodine and 10% potassium iodide. It naturally has this brownish-orange color. You can actually find a version of this right at your local pharmacy. This simple brown liquid is our ultimate chemical lie detector.
Section 3. The experiment procedure. Let's start the interrogation.
The Dr. Chem procedure for detecting starch is remarkably elegant. Step 1. Sample prep. We just take 1 milliliter of our suspect sample and drop it into a clean test tube. We also set up a control tube with just distilled water so we have a clear baseline. Step 2. We add the reagent. Using a pipette, we add 2 to 3 drops of Lugol's iodine into both tubes and give them a really gentle swirl. Step 3. We observe the color. We are waiting for an immediate visual transformation. And finally, Step 4. To totally prove the underlying chemistry, we run a thermal test by heating the tube in a water bath and then cooling it right back down.
Section 4. Observations and results. The confession.
Okay, here is the smoking gun. If there is even a trace of starch in that sample, again, down to 0.02%, that brownish-orange iodine instantly transforms into a striking, intense, dark blue-black color. It happens right before your eyes. Chemically, what you are seeing is the formation of a polyiodide complex. The polyiodide ions from our Lugol's solution are interacting directly with those helical chains of amylose we talked about, creating an adsorption complex that completely changes how the liquid absorbs light. It turns it from brown to an incredibly dark blue, sometimes looking almost totally black.
So how does this actually work at a microscopic level? Well, our sources tell us, quote, the triiodide and pentaiodide ions formed are linear and slip inside the helix structure. Remember when I told you to keep that spiral spring shape of amylose in your mind? Well, this is exactly why. When you mix potassium iodide with elemental iodine, you form these linear polyiodide ions. They're physically shaped perfectly to slip right into the hollow inner core of the amylose spiral. It is literally like a sword sliding flawlessly into a scabbard. Once inside, the electrons interact in a way that shifts their energy levels, absorbing visible light and creating that deep, deep blue hue.
It really all comes down to the geometry. The alpha-1,4 glycosidic bonds, linking the glucose molecules together in amylose, have this highly specific geometry that forces the whole chain to curl into a helix, with roughly 6 glucose monomers per turn. It beautifully illustrates the architecture of our trap. Now, obviously, the plant didn't design this to catch iodine. It designed it to pack energy as efficiently as possible. But the resulting geometry just happens to create the ultimate, perfectly sized trap for our iodine molecules, letting us run this incredible diagnostic test.
But wait, because step 4 has a literal thermodynamic parlor trick. If you take that dark blue-black test tube and heat it up in a boiling water bath, the color magically vanishes. The liquid turns transparent or pale orange again. Why? Because the heat energy physically shakes the amylose helix apart. The spring uncoils, the trap opens up, and the iodine just slips out. But if you take that exact same tube and run it under cold water, the starch molecules cool down, the spiral winds tightly back up, it traps the iodine all over again, and bam, the deep blue-black color instantly reappears. It's a completely reversible reaction, and it is absolutely mesmerizing to watch.
Section 5. Real-world starch applications. The verdict.
So, what does this mean for us outside the lab? Well, if you've ever had a sauce turn into literal glue or a pie filling turn to soup, it's because all starches are absolutely not created equal. Different plants have entirely different ratios of amylose to amylopectin, meaning their starches gelatinize and behave uniquely. Making gravy, you want cornstarch because it handles high heat well and gives a hearty, opaque finish. But if you're making a fruit pie filling, you definitely want tapioca. It gelatinizes at a lower heat and gives a gorgeous, glossy shine. Need a crystal clear glaze? Arrowroot is your guy. Making meals to freeze? Cornstarch actually breaks down in the freezer and weeps water, so you must use tapioca, which has awesome freeze-thaw stability thanks to that highly branched amylopectin network.
And this chemical variation totally changes the physical texture of baked goods too. Cornstarch creates a tender, soft crumb because its fine granules swell with moisture without adding chewiness. It's perfect if you want to melt in your mouth super delicate cake. Tapioca, on the other hand, yields a chewy, cohesive texture. That high amylopectin branching promotes elasticity, which is exactly why tapioca starch is a total must-have in gluten-free baking to stop your breads and cookies from turning into a crumbly mess. Swap one for the other without understanding the chemistry, and you've basically ruined dessert.
Oh, and one final, absolute game-changer tip for your kitchen. The chemical environment of your recipe heavily dictates your thickener. Arrowroot is fantastic for clear glazes, right? But if you put it in a dairy-rich sauce, the dairy proteins actively interfere with its gel formation, and your sauce breaks down into a slimy mess. Gross. Conversely, cornstarch is sturdy, but it weakens big time when combined with highly acidic ingredients like a rich tomato sauce or lemon juice. This basic chemical knowledge is quite literally the difference between feeling like a master chef and causing a kitchen disaster. So, case closed. We've profiled our polysaccharide, we've witnessed the dramatic blue-black iodine reaction in the Dr. Chem experiment, we unraveled the molecular spring of amylose, and we applied all of those findings to everyday cooking. It really leaves us with one incredibly empowering question. What other hidden chemistry is waiting right now in your kitchen cabinets? Seriously, the next time you thicken a sauce or eat a spoonful of honey, I really hope you see the brilliant coiled molecular structures working right there in front of you. Go grab some Lugol's iodine and start your own food investigation. Thank you so much for joining me on this explainer. Keep asking questions and keep learning.